Assalamu alaikum. You are watching Microbiology by Nouman. In this video, we will talk about coagulase test. Coagulase is an enzyme and coagulase test detects whether a bacteria carries coagulase enzyme or not. The aim or purpose of this coagulase test is to differentiate between Staphylococcus aureus, which carries coagulase enzyme, from coagulase-negative staphylococci, that means other staphylococci which do not carry coagulase enzyme. So coagulase test is used to differentiate between Staphylococcus aureus from coagulase-negative staphylococci, which are also known as cones. For example, Staphylococcus epidermidis or Staphylococcus saprophyticus. Staphylococcus aureus is coagulase positive. Talking about the principle of this test, coagulase is an enzyme that can convert fibrinogen into fibrin and resulting in a plasma to clot. So coagulase is an enzyme that converts fibrinogen into fibrin and results in a plasma to clot. Coagulase enzyme is one of the virulence factor of Staphylococcus aureus. That means it play a role in causing infection in humans or in animals. Staph aureus produces two types of coagulase enzyme, bound coagulase and free coagulase. Bound coagulase and the second one is free coagulase. Bound coagulase is also known as clumping factor. It is present or bound in the cell wall of Staph aureus. This bound coagulase is detected by slide coagulase test. Whereas free coagulase is an enzyme which Staph aureus secrete outside the cell. So it is secreted extracellularly and detected by tube coagulase test. So free coagulase is secreted extracellularly or outside the cell and detected by tube coagulase test. Slide coagulase test is a very fast test and this coagulase can be detected in 10 seconds, but this test can give us false negative results. That means the test was positive but declared as negative. Whereas this tube coagulase test is a definitive method for coagulase detection and can take up to 24 hours. Now we will discuss the procedure of slide coagulase test and tube coagulase test one by one. Slide coagulase test, we will take a clean glass slide and on this glass slide we will draw two circles A and B, and we will add few drops of water or normal saline to both of these circles. Then we will pick few colonies of bacteria and mix with these water, right? In the third step, we will add rabbit plasma to one of these circles, right? For example, here we are adding, in this B, we are adding rabbit plasma. Then we will rock the slide in figure 8 like motion, right? For example, this is the slide, we will rotate it like this way, right, for 10 seconds. Within the 10 seconds, we will look for clumping. Any degree of clumping it will be declared as positive. So in the very first step, mix bacterial colony with water. Add rabbit plasma. Roll the slide for 10 seconds and look for any clumping. Any clumping on the slide will be declared as Coagulase positive. That means when we see the slide, just like this one, a clumps can be visible, very clearly visible. That will declared as positive result, right? No clumpings mean that the result is negative. But this test can gives us false negative results. It's a very fast test, can be can be performed in 10 seconds, but this tube coagulase test is the definitive test that must be performed if slide coagulase test is negative. In tube coagulase test, while performing this test, we'll take two test tubes and in these two test tubes we will add rabbit plasma, 0.5 ml of reconstituted rabbit plasma. So this is the plasma. Similarly here 0.5 ml rabbit plasma. Then we will pick 2 to 3 colonies of suspected culture. Here we're taking staph aureus, which is Coagulase positive. So we will pick 2 to 3 colonies of staph aureus aseptically with sterilized loop or needle and mix with this plasma in one of these tubes. For example, if we're mixing plasma in this tube, right? And we will incubate these tubes at 37 degrees centigrade for 4 hours. Some people recommend it for first for 90 minutes, right? After 90 minutes or 4 hours we will see any sign of clumps that is formed in the tubes. If there is no clump seen, we will again incubate this tube at room temperature for 24 hours. After 24 hours we will see any kind of clumps, then the test is declared as positive. For example, if in this tube this clumps is visible, right, we will declare this test as positive, whereas in this tube we didn't add any culture, so this tube is negative, show no clumping. So write it down, the procedure: add 0.5 ml of rabbit plasma to each tube, pick 2-3 colonies of staph aureus or suspected culture and mix with plasma, incubate at 37 degrees centigrade for 4 hours. Look for any clump. If there is no clumps, again incubate for 24 hours. That's it from this video. If you have any question regarding this topic, you can ask in the comment section. Thank you.